In continuing with our second podcast on the skin and integumentary system, let's take a few minutes and talk about the cells of the epidermis. We can talk about keratinocytes, melanocytes, Langerhans cells, and Merkel cells. Now, the keratinocytes are highly specialized epithelial cells, and they're designed to separate the organism from its external environment. As we've said, the keratinocytes are the most numerous cell in the epidermis. Melanocytes are pigment-producing cells. During embryonic life, the precursors to melanocytes are derived from the neural crest. And this is going to be clinically very significant because neural crest cells, as you know, are migratory cells. And as you might imagine, tumors derived from melanocytes can be highly invasive. The melanocytes are located in the stratum basale. Langerhans cells are antigen-presenting cells. They're derived from bone marrow, actually from the monocyte precursors. And Merkel cells are associated with nerve endings or sensory cells in the epidermis. The origin of Merkel cells is actually not that clear. They have antigenic markers for both epidermal and nerve cells, and the Merkel cells are located in the stratum basale. Now, the stratum basale consists of a single layer of cuboidal to columnar cells. These are committed stem cells. They'll have a high mitotic index. The cells are firmly bound to a fairly substantial basal lamina. They're attached to each other by desmosomes, and they're attached to the basal lamina by hemidesmosomes. The cytoplasm of the cells in the stratum basale exhibits fairly substantial smooth ER, numerous free polyribosomes, and bundles of keratin filaments. The stratum spinosum consists of multiple layers of cuboidal to flattened cells. Most of the cells in the stratum spinosum are the keratinocytes, and as we've said, the keratinocytes are linked to each other by abundant desmosomes. You can see that in the middle image. You can see the spiny processes, or the prickle cells if you want. These are essentially the keratinocytes. There are masses of keratin bundles that fill the cytoplasm. Here you can see some of these intermediate filaments, and here you can see the desmosomes on this electron micrograph between the keratinocytes. The cytoplasm exhibits a smooth ER, free polyribosomes, and vesicles containing membrane coating granules, which are essentially glycolipids that are arranged in lamellae that resemble myelin. We'll talk about these a little bit later. Here's just an electron micrograph showing some of the keratinocytes. You can get a sense of these desmosomes that would be between the cells, and you get a sense of the spinous layer. Now, here you can see some dark pigment in the cells. These are actually melanin granules, and here showing these melanin granules as they collect over the nucleus of one of the cells. We'll talk about this a little bit later. Here are some of the desmosomes between the various keratinocytes. The stratum granulosum consists of 3 to 5 layers of oval to flattened cells. Again, mostly keratinocytes. In the light micrograph, you can get a sense that you can see some granules in these cells. Membrane coating granules are now very evident. That's what some of these granules are. They're being pushed toward the cell surface. Here you can see some of these membrane coating granules in the electron micrograph. As I said, they're being pushed towards the cell surface, and they're going to secrete into the intracellular space where they're going to form multiple lipid-rich water-repellent layers. There are also keratin granules, which are non-membrane bound, which contain histidine-rich proteins like filaggrin and trichohalin, and these are involved in holding keratin bundles together. The stratum lucidum is commonly seen only in thick portions of the palms and soles, and so the stratum lucidum would be an area right like this, part of the stratum corneum, right near the epidermis. This consists of several cell layers that are refractile. The cells in the stratum lucidum are going to have deteriorating organelles and nuclei. The cells are going to be packed with keratinocyte granules and keratin bundles. Now, on exams, I'll expect you to recognize the stratum corneum and thick skin, but I actually won't ask you to pick out the stratum lucidum. The stratum corneum, here you can see it on thicker skin or a thicker region of the epidermis like this, again consisting of a dead cell layer, and here the stratum corneum on a thinner portion of the epidermis. The stratum corneum consists of a few to many layers of organelle-free squames, of very flattened, thin cells. These cells are actually very wide. They may actually span 12 to 15 regular cell widths. They're essentially flat masses of keratin that are bound together by filaggrin and interspersed by lipid-rich layers that would contain remnants of membrane coating granules. The stratum corneum may be tightly packed, as shown on the top image, or it may consist of freely desquaminating layers. Here you can see some of the desquaminating layers on the bottom thinner image, but also the desquaminating layers on the top image on the thicker portions of the stratum corneum. Here's just an electron micrograph. These are layers of the dead squames. There's intracellular space, and here are some of these proteinaceous bundles that hold these squames together. Keratinocytes and the process of keratinization is really a process that very much relates to apoptosis. And this is a fairly complicated slide, so we're going to start out by describing it. So let's start by saying that cells in the stratum basale are going to divide to produce keratinocytes. As soon as the stem cells divide and produce the keratinocytes begin to undergo the process of apoptosis. Part of this process, though, the cells are synthetically active. They're synthesizing keratin filaments, and keratin filaments are given the name tonofilaments. And you may remember when we talked about epithelium, we talked about keratin as a cytochemical marker for epithelial cells. So these keratinocytes are synthesizing keratin. As the cells are getting more mature, they're going to enter the stratum spinosum. They're going to continue to synthesize keratin, and the keratin bundles are now going to group together into more substantial tonofilaments. As the keratinocytes move to upper layers of the stratum spinosum, they're going to synthesize keratohyalin granules, which are going to contain intermediate filament-associated proteins, the so-called filaggrins and trichohalins. They're going to also synthesize lamella bodies, which are membrane coating granules. And when the keratinocytes get to the upper layers of the stratum spinosum, they begin to initiate the final stages of apoptosis. Keratohyalin granules accumulate in cells of the stratum granulosum. The granule layer contents are going to be released within the cell, and that release of these granules is going to initiate the cornification process. And cornification involves breakdown of the nucleus of organelles and a thickening of the plasma membrane. And this is going to relate to a pH change. The pH is going to go from a neutral to an acidic pH, and membrane coating granules are going to be released, and that's going to cause a lipid barrier to form that's going to be a water type barrier. And then desquamination is going to occur as desmosomes between the cells break down. And this is going to involve a variety of serine peptidases that become active at relatively low pH. And I'm not going to ask you to remember these, but some of these are kallikrein-associated serine peptidases, and some of them are leukocyte-associated serine peptidases. This is just a schematic diagram, if you want, as well as with the electron micrographs. And these would be depicting cells in the stratum granulosum. So here is a plasma membrane of one cell. Here's a plasma membrane of another cell. And you might imagine multiple plasma membranes as these cells are stacking on top of each other in this area. But here are these lamella bodies that are being synthesized, and they're going to be fusing with the plasma membrane and releasing these high lipid content materials. They're going to begin to form a water type barrier, the so-called lipid envelope. And then there's a cell envelope that's forming within each of the squames of dead cells that are going to involve various lipids and intermediate filament type proteins like filaggrins and various various keratins. And the point I just want to make is the cell envelope and the lipid envelope consist of lamella body deposits and thickened plasma membranes. The plasma membrane is thickened by secretion of small proline-rich proteins as well as large structural proteins. Some of these are keratins, lauricins, involucrins, desmoplanktons, etc. You don't have to remember those. The important point is these lipids provide a water barrier and the proteins provide strength to that barrier. Keratinization is a self-regulated process of keratinocytes. The keratinocytes will synthesize epidermal growth factor that stimulates keratinization. They'll also synthesize and secrete transforming growth factor that suppresses keratinization. Stem cells in the stratum basale are mitotically active. They follow a circadian rhythm, so they tend to divide in the dark cycle. Keratinocytes are predestined to complete a 20 to 30 day life cycle, and then they're shed. Apoptosis begins almost immediately after they form. There are a variety of keratins that are made sequentially by the keratinocytes. There's at least 14 different types of keratins. I'm not going to expect you to know these, but for example, keratins 5 and 14 are made in the stratum basale. Keratins 1 and keratin 10 are made in the cells of the stratum spinosum. Different types of keratin and different mechanisms of keratinization yield structures like hair and nails. Here is just a diagrammatic image showing some other cells that are in the epidermis. So, for example, Langerhans cells can be located in the stratum spinosum, and the image on the right shows an immunostaining to a CD1 surface marker on the Langerhans cells. And you can see that the Langerhans cells, by this immunostaining, can be located throughout the epidermal layer. Here, melanocytes tend to stain clear with H&E stain. These are cells that are in the lower layers of the stratum basale, just in the very bottom layer of the stratum basale. Langerhans cells are antigen-presenting cells. They're stellate cells interspersed between keratinocytes in the stratum spinosum. They're a bone marrow-derived cell. We almost think of it as a monocyte-derived cell that's in the epidermis. They make up between 3 to 8% of the cells in the epidermis. They exhibit these rod-like granules here in the higher power shown like this, but here they're shown here. These are B-brick granules. We don't know the function of these granules. The Langerhans cells bind complement IgG and IgA. They phagocytize foreign matter. They can migrate to regional lymph nodes and present antigens to T cells. Langerhans cells are believed to be involved in contact dermatitis, in processes like inflammation, and in processes like psoriasis. And we're beginning to think that psoriasis may be the result of abnormal T cell activity. And let me just walk you through this slide. Recent studies suggest that psoriasis may result from an abnormal interaction or response of T cells to Langerhans cell signals. Essentially, foreign antigens enter the skin and will stimulate Langerhans cells. The Langerhans cells will migrate to local lymph nodes and activate cytotoxic and helper T cells. Once activated, these T cells will clonally proliferate and express a surface receptor called cutaneous lymphocyte antigen. The T cells are going to then proceed to the skin via the blood circulatory system, and this CLA is going to be recognized by selectins on the blood vessels in the endothelium. The T cells are going to diapedese and enter the epidermis. Here they're going to release tumor necrosis factor alpha and other factors which will cause the keratinocytes to release inflammatory factors and to undergo rapid mitosis. The inflammation is going to be due to increased lymphocyte activity. Now, the keratinocyte turnover rate, which is normally 30 days, is going to be rapidly increased, so they're going to turn over in 3 to 4 days, and as a result, keratinization is going to be incomplete when the cells slough off. Now, why antigens cause the abnormal T cell response in different individuals is not really clear. Merkel cells are innervated stellate cells of the stratum basale. They may be a modified keratinocyte or a specialized nerve cell. The cytoplasm of Merkel cells contains dense core granules. Here you can see them at a little bit higher magnification. These granules resemble those granules that you see in endocrine cells, but we don't know the function of these granules in the Merkel cell. Merkel cells have abundant sensory areas, so they'd be very abundant in the fingertips. They're thought to be mechanoreceptors. Now, this is an interesting sidelight. Merkel cell carcinoma is very rare, but it is a very highly aggressive form of skin cancer. Melanocytes are stellate cells in the stratum basale. They're neural crest-derived cells. The differentiation is regulated by PAX3 genes, and one melanocyte usually maintains an association with a fixed number of keratinocytes, and usually you see the melanocytes in the stratum basale, and usually about every 10th cell is a melanocyte. And in a routine histological section that is stained with H&E, you can see these large Claire cells in the stratum basale. Now, under stimulation of UV light and under the influence of melanocyte stimulating hormone, melanocytes will synthesize melanosomes. And melanosomes are membrane-bound vesicles that contain melanin granules. Now, melanin itself is produced in cells that contain tyrosinase activity. It turns out that skin color is dependent on tyrosinase activity and on degradation of melanin. Keratinocytes in light-skinned individuals exhibit more melanin degradation. There are actually no differences in the number of melanocytes between dark-skinned and light-skinned races. Now, skin pigmentation might also relate to age, to gender, to hormone levels, perhaps to genetic and environmental factors, and there are some chemical differences in melanin pigments. Now, I do want to make this point. Pieces of melanocyte cytoplasm containing melanosome vesicles are going to be released by cytokine secretion from the melanocyte, and then they're going to be phagocytized by neighboring keratinocytes. That process is called pigment donation. And then once the melanin granules get into the keratinocytes, they're going to be placed above the nuclei, and it's thought that that occurs to protect the DNA against UV light. This image shows melanocytes in the stratum basale. Here happens to be a Langerhans cell, but here's the melanocyte. Here it is at the electron microscopic level. Note that the melanocyte has all these processes, so sometimes it's called a stellate-shaped cell. These processes are going to bud off melanosomes by a cytokine secretion, and these melanosomes are going to be taken up by the neighboring keratinocytes, and that's shown on the bottom image here. So they have these pre-melanosome formations, the synthesis of melanin in the early melanosomes, and then the melanosome secretion. And then what this is showing is melanosome degradation, and you can note that in light-skinned individuals more melanosomes are degraded as opposed to individuals with darker skin. Here you can actually see some of the melanosomes in the melanocyte. Here are the melanin granules as they've been incorporated into the keratinocytes, and you can see these melanin granules that are clustering over the nucleus. And again, this is thought to provide some UV protection for the DNA in the nucleus.